All right, welcome. So I want to quickly review digestive physiology, and these are the learning objectives, the kinds of things you should know, the classes of carbohydrates. That's um, from complex starches or polysaccharides all the way down to monosaccharides, and identify the structures that make amylase that helps to break that down. Um, and also things like how acidity in the stomach might affect amylase, how what form of the carbohydrates are absorbed. So please go through these learning objectives pretty, um, pretty in a detailed manner. Be sure you can answer these questions. Same thing for proteins. How are the enzymes activated or inactivated? Where do they work? And in what form is the protein eventually absorbed? And finally, lipids. Again, the same thing. Um, and you will be required to interpret the results of the experiments you performed in lab. So first, digestion is both mechanical, the chewing of the food, the churning of the food in the stomach, and so on. And there's the chemical digestion, which is done by either enzymes or even the effect of chemicals, inorganic chemicals like hydrochloric acid that help to denature proteins and things like that. So there's a chemical part, which again is two parts. There's the part by inorganic substances, but the most of it is done by the by the enzymatic digestion. And then another term you should remember what that means. Secretion means whatever is being made is being released into the lumen. So a goblet cell will secrete mucus into the lumen, okay? And absorption is the opposite direction. So first off, a little caveat, there are more enzymes that I mentioned in this lab. So I would say restrict yourself, if you go back to read the textbook, to the ones we covered in lab. But of course, for me, no knowledge is lost. It's always good to have the bigger picture of things. All right, so carbohydrates, three classes: monosaccharides or simple sugars, glucose, fructose; disaccharides or disaccharides, sucrose, maltose. Sucrose is one glucose and one fructose. That's table sugar. Maltose, for example, is two glucose molecules fused together. So as you can imagine, the major difference here is the size. Remember, I mentioned, or let me mention it now, that monosaccharides are typically around about 180, perhaps 200 molecular weight units. Disaccharides are twice that. The larger a molecule is, the harder it is to cross the membranes, particularly water-soluble molecules like carbohydrates. So that tells you that ultimately it's the monosaccharides that are the form that are mainly absorbed across the epithelium in the small intestines. And then polysaccharides are things like starch. So how does it digestion of carbohydrates? So first of all, you have your salivary amylase that helps to begin that process. So at best, it might convert some branches of a complex polysaccharide to single chain branches. So hear me out. I'm not saying it makes monosaccharides. It might, but majority of the monosaccharides are made further down in the small intestines. So all it might be able to do in the little time that the food spends in the mouth, assuming, of course, you chew your food 30 times, it breaks the branches into straight chain oligosaccharides, that is long chain simple sugars. Excuse me, long chains of simple sugars. And then, but of course, by the time that food gets into the stomach, the acidity of the stomach will inactivate that. And I believe we did that experiment in the lab. We'll get to it. But the bigger chunk of this is done by the pancreatic amylases. If you notice, in the pancreas, the pancreas secretes its enzymes or its entire cargo into the small intestines, and that's the largest part of the small intestines. Even though there's a large intestines, that's only about the size. in terms of area, the largest area is in the small intestines because of the folds, the villi, and the microvilli that are in that section of the stomach. Okay, and so the pancreatic amylases have a much longer exposure to the carbohydrates, and so they do the heavy lifting here. And so eventually it breaks the straight chain oligosaccharide chains that were formed from the mouth now begins to break those down into smaller units. It could even go as far as simple sugars like glucose itself, or so that's glucose, or it could be disaccharides like maltose. Okay, and then finally, for most of the disaccharides, they are now broken down by specific enzymes that are at the brush border. And so those would break your maltase. The maltase will break down maltose into two glucose molecules, smaller and easier to absorb. Lactose will do lactase will do lactose to glucose and galactose, and so on. And so this is what that looks like. So that's one maltose gives you two glucose molecules, and then this can cross the brush border. Oh, that's a poor brush border, but you get my point. And final product, monosaccharides, and these are absorbed eventually across the intestinal epithelial lining. Proteins, so your fish, your steak, things like that, are large and long. Strands of amino acids. Okay, and the amino acids are linked by peptide bonds. So where does digestion of protein start? This starts with, number one, it starts in the stomach and with the help of hydrochloric acid. So hydrochloric acid will begin the process of denaturing your proteins. And so that denaturing facilitates chemical digestion. The other thing it does is hydrochloric acid will activate pepsinogen. So from pepsinogen to pepsin. And pepsin is active, it will break peptide bonds. That's what it does. And again, you now form a polypeptide and some, dipeptides and tripeptides and oligopeptides in general from the complex proteins. And then once that leaves the stomach, it goes to the small intestines. And of course, the pancreatic juice helps to add to it trypsinogen. So it has trypsinogen in it, which once it gets into the small intestines, the enteropeptidase, which is synthesized in the small intestines, in the duodenum, will activate trypsinogen to trypsin. Trypsin is active, it will begin to break down peptide bonds extensively. Ultimately, you begin to form a lot of amino acids as well as dipeptides, which is two amino acids. And so this is what that looks like, a bunch of di and tetrapeptide, a dipeptide here, a dipeptide there. So now you have this dipeptidases that will break those down to give you individual amino acids. And again, these individual amino acids typically have a molecular weight of around 200, give or take. And that can be much easily absorbed compared with dipeptides or tripeptides. Okay. Next is lipids. So lipids, they are, of course, they're not water soluble. Two types: cholesterol, which do not need to be broken down to be absorbed, and then there's triglycerides. Triglycerides are made up of glycerol and three fatty acids. So the bond of the fatty acids with the glycerol is what gives a glyceride. So triglycerides, three fatty acids. And these have to be broken down. But for this to happen, several things are needed. First is the lipids. First of all, there's the lingual lipase, which is inactive. It is inactive. It's in the from the mouth and from the salivary gland. That's inactive. Okay, I'll come back to triglycerides later, but and then once the lingual lipase gets to the stomach, the hydrochloric acid in the stomach helps to activate the lingual lipase. So if you add that to the two other effects of hydrochloric acid that were described before, that's another key function or key role of the hydrochloric acid. The other two that I mentioned before were that it helps to denature proteins, it helps to activate pepsinogen to pepsin. In the same way, it helps to activate lingual lipase. Okay. And then you also do have gastric lipase as well. So if you think about it, together, gastric lipase in the stomach and lingual lipase digest about 30% of triglycerides. So that's quite a load. That's significant. And then once the triglycerides get to the small intestines, the bile from that is made in the liver but stored in the gallbladder is released, and it meets with the pancreatic juice at the at the sphincter or the ampulla actually, and it's called the sphincter of Oddi. But the point, you don't need to know that. But at the point where the pancreatic duct and the bile duct come together, everything is mixed. So now you have bile as well as the pancreatic lipases. Okay. Now, what's important here is regarding triglycerides, the bile helps to emulsify, that is, break up the fat, because fat and water do not mix. You know that. It breaks it up into small molecules or small particles. Okay, so I'll mention this. Whenever you use what people call a hydrating body cream, that creamy nature really is a formation of many tiny micelles, actually, whereby the micelles, the oil droplet is surrounded by tiny water droplets, and that's what makes it such that that cream that you use, that hydrating cream, does not separate into oil and water, which will be unsightly for a cosmetic product. So essentially, that's what bile does. It helps you to break down the oil into the lipid into small droplets, and each of those droplets will now be surrounded by water. It allows us to have a creamy suspension of lipids. Okay. Excuse me. So that's what's called emulsification. Now, because after you emulsify, that enables the lipases to have better contact with the triglycerides and begin to break down those bonds. Okay. And so that's what it says here, where the pancreatic lipase released at the same time emulsifies the fats into monoglycerides and 2 fatty acids. And it's in this form that the fatty acids are now absorbed. Once they're absorbed, they reconnect back again to form triglycerides within the intestinal epithelial cells. But don't worry about that. There are mechanisms to help shuttle the reformed triglycerides out of the epithelial cell into the interstitial space, which then goes into the lymphatic vessels called the lacteals. Okay, all of this is not on the slide, but hopefully you can go back and just understand that. And so that repackaging, ultimately, whatever you absorbed you absorb in the stomach and goes to the lymphatics, goes to the veins eventually, goes to the liver, and the liver reprocesses that and makes them, conjugates them with proteins. And we now begin to talk about VLDL, very low density lipoproteins. That's because now those triglycerides and cholesterol have been packaged with proteins to enable them to float around in blood. Because you no longer have micelles at this point. The micelles would have been dissolved after after they've been absorbed. The triglycerides have been absorbed into the intestinal epithelium. But too much detail. You don't need to know all of that. But sufficient to know how, for example, the bile helps the digestion of triglycerides. And so you have those two things, and these are absorbed into the epithelial cell, what I just described. And in the epithelial cell, again, what I just described, the triglycerides are reformed and they are wrapped to the protein to form the chylomicrons. And those are exocytosed into the lacteals and therefrom enter into the blood. And the blood takes it to the kidneys and all of that, to the liver and all of that good stuff. And that's a picture right here on the right. Okay, so there's a sheet of paper that is in the supplemental material that you can use to learn this thing. What this thing really does, and I highly recommend you use it for studying, is it tells you at which stage. So if you notice, this is kind of down as you go from the mouth, saliva, to the gastric, to now you are in the pancreatic fluid. So now you're in the small intestines right here, and you have this fluids, and so it kind of tells you how things happen and what happens at each stage. So for example, salivary amylase is in the oral cavity. It breaks down polysaccharides, and you get short chains of starch. And then lingual lipase in the mouth helps to do some digestion, so you have diglyceride from triglyceride and maybe one fatty acid, and so on and so forth. And at the end of the day, proteins end up as amino acids, carbohydrates end up as monosaccharides, and fats end up emulsified in micelles. And again, the actual proteins that help to break down the actual enzymes that help to break down proteins that you need to know, carbohydrates, and for lipids, what each of these do. Okay, and what's formed at the end of the day. So there you go. So, okay, we'll ignore that one, but excuse me. Let's get to, so this is that sheet that is in your additional or supplemental materials link on Blackboard. So you can use this to kind of test yourself. Try to fill out as much of this as you can to be sure of what you know. Don't just go off of the answers that I just displayed. Another thing is really quickly, okay, this is a reminder for you of what you did in lab and what each experimental condition was for the protein experiment. Okay, so for the protein experiment, so you had here, look at the answer key. So that is water at the top. So that's water. Okay, and this is trypsin. Okay, and note the temperature. And this one had no substrate, so this is basically your negative control. Negative control, because there was no substrate. Now the substrate here is BAPNA. Notice there's BAPNA here. There's water, but no enzyme. So this is also a different kind of negative control. And here is um, again, here is BAPNA and trypsin at 37 degree, BAPNA and trypsin at 4 degree. So so you can go back to your lab notes and see what you found under those conditions and be able to explain that. Um, Same thing, the lipid, just follow this. This should guide you. This sheet of paper is also in your supplemental material.